The quest to understand life and perhaps even to create it is not entirely new. In the dawn of the 20th century, a new field began to stir. Visionaries imagined a world where biology could be engineered. They saw life not just as something to be observed, but as something that could be designed and built. These were bold ideas born from a growing understanding of the cell and chemistry. The laboratory was becoming a place of construction. Scientists began to think of biological components as parts to be understood and assembled. This was a radical departure from traditional biology. The seeds of a new creation story were being sown in scientific endeavor. The question of life's origin has captivated humanity for millennia. How did the first living cells arise from non-living matter? In 1952, a remarkable experiment sought to shed light on this profound mystery. Stanley Miller and Miller-Urey at the University of Chicago designed an apparatus to simulate the conditions of early Earth. They imagined a world with a reducing atmosphere, filled with methane, ammonia and hydrogen, all above a vast ocean of water. This was their attempt to recreate the primordial soup, the very cradle of life within the confines of their laboratory. Water was heated to produce vapor, mimicking evaporation from ancient oceans. This vapor mixed with the gases, and then, crucially, electric sparks were discharged into the mixture. These sparks represented the lightning storms thought to be common on the young Earth, providing the energy needed for chemical reactions. The mixture was then cooled, causing condensation, and the resulting liquid was collected. After just 1 week, the clear water had turned a murky reddish-brown. Analysis of this liquid revealed something extraordinary: amino acids. The Miller-Urey experiment demonstrated for the first time that complex organic molecules could be formed spontaneously from simpler inorganic precursors under early Earth conditions. The journey from understanding life's components to actually building life took a monumental leap forward in the early 21st century. At the forefront of this was the pioneering work of Craig Venter and his team. In 2010, they announced a breakthrough that resonated around the globe. They had successfully created the first self-replicating synthetic bacterial cell. This was not merely modifying an existing organism, it was constructing a genome from scratch using chemicals off the shelf and then booting up this synthetic genome within a recipient cell. Scientists first sequenced the genome of a naturally occurring bacterium. Then they chemically synthesized this entire genome piece by piece in the laboratory. This synthetic DNA, over 1 million base pairs long, was then carefully assembled. The next challenge was to transplant this man-made genome into a different bacterial cell, one whose own DNA had been removed or disabled. The cell with the synthetic genome, nicknamed Cynthia by some, began to replicate. It produced new proteins and divided to form daughter cells, all under the direction of the artificially created DNA. Craig Venter famously stated, "This is the first self-replicating species we've had on the planet whose parent is a computer." It demonstrated that the code of life could indeed be written and brought to life. This breakthrough opened up vast possibilities for synthetic biology. While synthesizing entire genomes marked a giant leap, another revolution was brewing, one that offered unprecedented precision in altering existing life forms. This was the advent of CRISPR-Cas9 gene editing technology. Emerging prominently in the early 2010s, CRISPR transformed the landscape of genetic engineering. It provided scientists with a tool that was remarkably simple, incredibly efficient, and far more precise than any previous gene editing methods. It was like discovering a molecular scalpel that could find and alter specific sequences of DNA within the vast complexity of a genome. CRISPR-Cas9 is based on a natural defense mechanism found in bacteria. Bacteria use it to fight off invading viruses by cutting up the viral DNA. Scientists ingeniously repurposed this system. The CRISPR part acts like a guide, homing in on a specific DNA sequence within a cell's genome. The Cas9 enzyme then acts like molecular scissors, making a precise cut at that targeted location. Once the DNA is cut, the cell's natural repair mechanisms can be harnessed to make changes, to delete a gene, insert a new one, or correct a faulty sequence. The impact of CRISPR has been felt across all areas of biology and medicine. In research laboratories, it has accelerated the study of gene function, allowing scientists to quickly understand the roles of different genes in health and disease. In medicine, CRISPR offers potential new therapies for genetic disorders. Beyond modifying existing life or synthesizing known genomes, another fascinating frontier in synthetic biology is the creation of protocells. These are not yet fully living cells as we know them, but rather simplified, artificial structures that mimic certain characteristics of life. Researchers in this field are attempting to build life from the bottom up, starting with basic chemical components. They seek to understand how the earliest, most primitive forms of cellular life might have emerged on Earth, and in doing so, perhaps create entirely novel, minimal life forms in the laboratory. Protocells are typically designed to have a few key features. They often possess a membrane-like boundary, separating their internal environment from the outside world, much like a cell membrane. Inside this boundary, they might contain simple genetic material, perhaps RNA or a synthetic equivalent capable of storing information and even replicating. Some protocells are also engineered to exhibit basic metabolic activity, taking in food from their surroundings and converting it into energy or building blocks. The goal is to create a system that can self-maintain, self-replicate, and even evolve the fundamental hallmarks of life. The research into protocells is incredibly diverse, with scientists exploring many different approaches. Success in creating truly autonomous, evolving protocells would be a monumental achievement. It would not only provide profound insights into the origin of life on Earth, but could also lead to new biotechnologies. The power to create and modify life is one of humanity's most profound achievements, yet with this power comes immense responsibility. The ethical implications of synthetic biology are vast and complex. We must navigate a labyrinth of moral questions. Biosafety and biosecurity are immediate concerns. How do we ensure new organisms do not escape and cause harm? Robust safety protocols and international oversight are crucial. Beyond safety, there are deeper philosophical questions. What is the moral status of artificially created life forms? If we create sentient organisms, what rights should they have? Editing the human germline raises concerns about altering evolution. We must balance knowledge with respect for life and ethical principles, proceed with courage and caution, mindful of our actions' consequences. While the promise of synthetic biology is immense, the path to realizing its full potential is fraught with significant scientific and technical hurdles. Creating life, or even substantially re-engineering it, is an extraordinarily complex undertaking. Life as we know it is the product of billions of years of evolution, resulting in intricate and finely tuned systems that we're only just beginning to fully comprehend. One major obstacle is our incomplete understanding of fundamental biological processes. Even for the simplest organisms, we do not fully know the function of every gene or how all the cellular components interact to produce life. When scientists design synthetic genetic circuits or attempt to build a minimal genome, they often encounter unexpected behaviors or failures because of these knowledge gaps. Another significant challenge lies in the sheer complexity of assembling and controlling biological systems. Synthesizing long strands of DNA accurately and efficiently is still a technical feat. Getting these synthetic constructs to work reliably within the context of a living cell with all its existing machinery is even harder. Biological systems are inherently noisy and variable, unlike the precise and predictable behavior we expect from electronic circuits or mechanical devices. Developing robust and predictable biological parts and chassis that function reliably across different conditions remains a key area of research. The journey is long, but the pursuit of knowledge drives us onward. The future of synthetic biology stretches the imagination to its very limits. We are moving towards an era where life itself can be programmed. Imagine microorganisms engineered as tiny biological factories. These organisms could revolutionize industries and tackle environmental problems. The potential extends beyond Earth. Synthetic biology could play a crucial role in space travel and colonization. Organisms could help create breathable atmospheres on Mars. They might produce food and recycle waste in space. Synthetic biology could turn barren landscapes into cosmic gardens. The journey into synthetic biology, from its earliest conceptual whispers to the tangible creation of synthetic cells and the precise editing of genomes, is a testament to human ingenuity. We stand at a remarkable juncture in history, wielding tools that allow us to read, write, and even rewrite the code of life. This burgeoning field is rapidly reshaping our understanding of biology. The implications for medicine, agriculture, industry, and our environment are transformative. This power, however, is a double-edged sword. With the ability to create and modify life comes an immense ethical weight. The potential for unintended consequences, misuse, or the exacerbation of inequalities cannot be ignored. We must engage in thoughtful dialogue and establish robust ethical frameworks. The awe inspired by our innovative capacity must be matched by a deep humility and respect for life. The innovations in synthetic biology compel us to confront fundamental questions about life. What does it mean to be alive? What is natural and what is artificial? And what is our role as a species that can now actively participate in the evolutionary process? By embracing innovation with a strong ethical compass, we can harness the profound power of synthetic biology.